Hello everyone. For my VoiceThread project, I'll be reviewing the 2019 article by Eshtat et al. That is titled Phase 1/Phase 2A Clinical Trial of Gene-Corrected Autologous Cell Therapy for Recessive Dystrophic Epidermolysis Bullosa. To start, I would like to warn you that some of the images seen are graphic, but viewing the wounds truly puts an emphasis on why treatment for epidermolysis bullosa, or EB, is necessary and would help patients drastically. EB is a family of dermatological diseases and occurs when the body does not produce a protein called collagen 7. Recessive dystrophic epidermolysis bullosa, also known as RDEB, is a subcategory of EB that is inherited from patients that are carriers of EB. RDEB patients have a lifetime of painful symptoms, which include their skin being very fragile, leading to extreme blistering and skin loss that occurs with very little friction. These wounds affect external and internal skin, being equivalent to third-degree burns. Blisters can form in very uncomfortable areas. For example, some places include the surface of the hands, eyes, mouth, esophagus, armpits, legs, and even the anal sphincter. Unfortunately, patients have to deal their whole life with symptoms and continuous wound care. Before the age of 35, they usually develop an aggressive skin cancer called squamous cell carcinoma, and these are in locations of heavy scarring and chronic wounds. Patients with RDEB have mutations in the COL7A1 gene that result in the inability to produce functional type 7 collagen proteins, or called C7. Collagen 7 is a triple helical protein that has two non-collagenous domains, NC1 and NC2. Along with this, it has forms of anchoring fibrils. In the majority of RDEB patients, the NC2 domain is not present. Collagen 7 acts as an anchor to the skin, attaching the two layers of skin. In patients without C7, sheets of skin are torn off by any type of friction that forms blisters and lesions. All these symptoms affect everyday activities on a daily basis for RDEB patients, and if you battle long enough, it becomes fatal. There are currently no treatments other than normal wound therapy procedures like bandages and antibiotics for infections. Simple baths for these patients end up being very painful acid washes that clean out open wounds. The quality of life for these patients is horrible. Any type of treatment would help tremendously. Thankfully, research is being conducted to find a possible method to treat patients. This single-center Phase 1/Phase 2 study follows RDEB patients who received gene-corrected autologous cell therapy and examines the long-term efficacy, safety, and patient-reported outcomes. This clinical trial had 7 participants enroll and began in October 2013 through March 2017, where the last patients were treated. These patients were considerably young, between the ages of 18 and 45. With wounds that were about 1 to 20 years old. All of them experienced other RDEB symptoms like corneal erosions or anemia, and one female had a history of squamous cell carcinoma at the age of 32. Patients chose their 6 worst wound sites, in which 5 of them were treated and 1 was left as a control. Patients selected their most problematic wound sites to receive treatment and were immobilized for 2 to 8 days. All patients had negative C7 antibodies and no mutations were formed. Researchers in this trial isolated skin cells from participants and then cultured the cells. The corrected human COL7A1 gene was then transfected into these cells and were organized into epidermal sheets before grafting onto the patient's wound sites. To deliver the corrected gene, the vector used was LZRSC retrovirus, also known as the gibbon ape leukemia virus. Researchers used visual inspections to identify if the wound had any accelerated healing or if this treatment would even work. Biopsies were obtained from patients, and by using IEM and IF techniques, they searched for any anchoring fibrils in the skin sample. Lastly, serum was extracted from all 7 participants to know for any anti-C7 antibodies. This phase 1, phase 2 study was conducted to ensure the safety of this particular gene therapy and make sure this type of treatment does not lead to oncogenesis to promote tumor proliferation. Along with this, researchers wanted to see how long collagen 7 lasted in patients and if any antibodies were formed against C7 due to autoimmune responses from the gene therapy. This is because the body could identify the corrected gene as foreign and target that protein all throughout the body. The results from this trial was considered a success because at 6 months, 95% of all wounds were healed by at least 50%, where control sites had no wounds 50% healed. At the 12-month mark, some wounds began to get worse, with only 68 healed compared to 17%. At 2 years, the treated area saw 71% recovery, and the control stayed at about 17%. At this point, it can be noted that participants that became stable after 2 years tended to stay stable. Although there is long-term data necessary for patients 5, 6, and 7, the overall results were promising because it showed that gene therapy was successful in most wound sites for patients. This image shows the progression of different wound sites from baseline throughout the 24 months. The top images show participant 7's back, where D, C, and A are the graft sites placed by the researchers. At baseline, it looks horrible, but at 2 years, you can see the whole new layer of skin that is scarred over the wound. Letters B and C are both pictures from participant 5. In B, the right arm was used as the control site, illustrating natural healing progression with no significant improvement. C shows the left arm and where grafts A and B were placed. At months 3 and 6, the wound has recovered and the skin grafts are doing well. At 12 and 24 months, lesions begin to form again, but the wound site has drastically improved compared to baseline. These graphs show the percentage of wound healing comparing treated versus untreated wounds. Graph A shows the percentage of wounds that have recovered 50% or greater. It can be seen that in the treated group, at 3 to 6 months, 36 out of 38 wounds were healed, where the untreated wound site at 3 months had 2 out of 6 and none at 6 months. Similar results were seen when looking at the 12 and 24 month mark. About 26 out of 38 wounds were healed, where about only 1 of 6 for untreated wound sites. Graph B shows wounds that have healed 75% or more, illustrating how the best results are at the start of treatment. Then 6 to 12 months, it decreases, and at 24 months, it rebounds, improving wound sites. This image shows a comparison of skin biopsies that were isolated from participant 4. When looking at the 2-year picture of letter C, there are small black dots that illustrate the localization of anchoring fibrils. This was done by immune electron microscopy, and it's seen that these dots do not exist in the baseline. Image D uses immunofluorescence to better show where the fibrils are located in the layers of skin. The majority of results in the study are visual and measure healing progression, but scientists also noted the molecular correction of NC2 and AF in all participants. At 3 months, all participants had elevated levels of NC2 and AF, with the exception of participant 1. 6 to 12 months is where there's a drop-off to these levels. The research is still being conducted for some patients, and more research can help provide a better understanding in terms of molecular correction. RDEB patients that survive long enough are susceptible to squamous cell carcinoma, or SCC. In this study, only participant 5 previously had a history of SCC. Sadly, participant 5 and another subject developed SCC over the span of 2 years. Researchers in this study wanted to understand if the retrovirus LZ-RSC was responsible for the cancer growth. Biopsies were taken from the tumor and PCR tests were conducted. These tests showed that LZ-RSC was not found in the tumors and SCC was not local to the skin grafts that were placed. Scientists took this data and interpreted that oncogenesis did not occur directly because of LZRSC. The serum taken from all patients was analyzed to detect antibodies against C7. Only trace amounts of IgG was noted in 5 patients, but researchers identified high levels of IgG in 2 participants. Patient 4 was the only one that had these antibodies at one of the wound sites. Researchers wanted to test if the response was systemic or if it was localized. They saw that in 2 patients that had high levels of IgG, it was localized to the wound site. Because these participants were unable to produce the NC2 domain, researchers in this study argued that this was not an autoimmune response, but more of an immune response to the foreign protein. In conclusion, this phase 1, phase 2 clinical trial was considered successful because it provided vital data for future research in this field. There's continued research required for the immune response to NC2. Although molecular corrections are important, post-operation wound care can be vital for the healing process. Some wound sites were harder to treat and did not provide the best results because of mechanical factors. For example, open wounds on your arms, leg, chest, or back have less healing due to exposure to movements when conducting daily activities. Overall, the data showed that treated wounds had a rate of healing that was 6 times greater than the control. Phase 3 is underway and is currently recruiting with 11 participants, and April 2022 is scheduled as its completion date.